Welcome to the Curiox Biosystems webinar. We have 3 speakers for you today. We will begin with Dr. Chan-Ying Ke, who will give an overview of the Laminar Wash system. She will be followed by Bertrand Yeung from BioLegend, and then Antonino Montalbano from Immunize. The presentation will last approximately 30 minutes. Please type your questions in the question box, and we will get to them at the end of the session. A recording of this webinar will be available shortly after at www.curiox.com. And now, Chan-Ying Ke.
Thank you for the kind introduction, Sheila. Welcome to our joint webinar. We are excited to share customer results with you. Our main speakers, Bertrand and Antonino, will follow me and will share experiences and data from the proteogenomics and multi-omics experiments prepared using Laminar Wash. Many who perform research with single-cell sequencing and proteogenomics will immediately see the value of Laminar Wash technology in their work. Many researchers have taken the centrifugation step for granted, as until recently, it has been the only method to remove excess reagents from suspension cells. But have you considered what centrifugation might do to your cells? In this image showing cells expressing red fluorescent protein, or RFP, spiked in with non-fluorescing cells, samples washed by centrifuge are observed to be lower in count, distorted in morphology, and importantly, have lower expression of RFP. This can be contrasted with cells that did not undergo centrifugation. The cells appear viable and are significantly brighter in red fluorescence. Now looking at the big picture, the red fluorescent protein in this image can represent any protein of interest in your cells, be it chimeric antigen receptor or CAR, checkpoint blockade proteins, tumor antigen, or that elusive biomarker that is sensitive to mechanical handling. It is now possible to wash your cells to reduce non-specific background staining while maintaining fidelity to protein expression using Laminar Wash. The problem of non-uniform cell loss in centrifugation is also a well-known issue frequently discussed on research forums. This leukocyte staining from whole blood showed that neutrophil and myeloid cells are poorly retained or stained in the centrifuge sample, but the same populations are prominent in Laminar Wash prepared samples. This result has been reproduced in other flow and mass cytometry experiments showing loss of cells specifically in the myeloid compartment. Next, I'm going to show a video clip of how the Laminar Wash system runs. We have 4 sets of cells in triplicates on the Laminar Wash 96 plate. The sets of triplicate wells contain blue food coloring solution mixed with green fluorescent beads only, and plain buffer. After 20 minutes of rest, the beads settle on the plate just like cells would. The user then places the plate on the washer platform and hits the start button. The plate goes in and the fluidic head comes down to the plate. Here is a close-up of the plate during the wash. The fluidic head provides 2 nozzles per well, one for dispensing fresh wash buffer and the adjacent one for aspirating spent buffer. During the 2-minute operation, it performs 7 cycles of dispensing and aspiration, diluting the reagents and antibodies exponentially with each cycle. When the wash is completed, the plate is returned, and you can see that the food coloring solution is completely gone. The food coloring represents tagged antibodies, while the green fluorescent beads, which would in this case represent cells, are retained on the plate. Now you may wonder how come the coloring was washed away while the beads are retained. The beads are not attached to the plate by any interactions, just as cells aren't. I'll show you in the next slide how this works. The retention of beads and cells on the plate is based on well-characterized forces of gravity and laminar flow, which are very well-known forces in nature. The secret is the use of each force on an almost flat Wallace plate, so we control the forces. The cells are mixed with antibodies on the plate and are settled by gravity over standard incubation periods during a staining protocol. Once the cells are settled, the action of dispensing and aspiration at low flow rate above the cells doesn't disturb them. The wash solution forms a laminar flow during dispensing and aspiration and minimizes turbulence of the fluid flowing due to lateral fluid movement. The velocity of the fluid becomes 0 at the surface of the plate, and at the very bottom of the plate where cells are located, cells are not subjected to high flow rates of the wash buffer. And over this thin fluidic layer, Washing happens by diffusion rather than turbulence of the fluid, and this is how we can wash the cells without losing them. Interestingly, when we perform an identical dispensing and aspiration in a conventional wort plate, the action of dispensing and aspiration causes turbulence, which will lift the cells from the bottom of the plate, leading to cell loss. Our users have experienced multiple benefits of adopting Laminar Wash and eliminating centrifugation from their workflow. In flow cytometry assays, the high cell retention allowed users to evaluate markers and functional responses on rare cell populations. The increase in viability and removal of debris improved resolution, particularly in primary tissue preparations. In single-cell genomics and proteogenomics studies, greater cell retention gave confidence in reflecting native tissue composition and expression, while reducing non-specific background from sticky nucleotide-based reagents. Bertrand and Antonino will be elaborating further on their observations in these aspects. The compact footprint of Laminar Wash washes allows complete cell preparation workflow within the biosafety cabinet and reduces aerosolization of infectious samples caused by centrifugation. Laminar Wash systems are also amenable to partial workflow automation, as shown here with a thermal orbital microplate mover, or complete automation with our Auto 1000 robotic system. Replacement of the centrifuge simplifies the automation setup, making automation now more affordable and compact. The Laminar Wash family of systems has a variety of throughput and automation level to suit every lab's workflow and budget. The compact size Mini has an 8-well washing throughput from a 16-well strip and is popular among mass cytometry and single-cell sequencing users. For higher throughput and streamlined workflow, the HT2000, shown here with an LCD touchscreen interface and an optional automated buffer exchanger, processes 96 wells at once. The Auto 1000 integrates a HT2000 and buffer exchanger in a Hamilton Nimbus automated liquid handling system. It comes pre-programmed with modifiable protocols that are easy for the user to immediately adopt with no additional scripting needed. The platform is designed to produce the most quantitative and reproducible results by reducing user variability and day-to-day variation prevalent in manual sample handling. Unlike custom automation or centrifugation-based systems, the Auto 1000 provides easy turnkey automation and exceptional flexibility. In addition, the Auto 1000 is much more compact and affordable and lower maintenance than automation systems built around centrifugation. Here, I would like to quickly illustrate how simple and intuitive it is to transit from conventional centrifugation workflow to Laminar Wash using the Mini 1000 and a simple staining protocol as the example. The Mini 1000 is extremely compact and can be easily set up in a biosafety cabinet. Prepare Laminar Wash 16-well strip by placing in its washing adapter. Cells can be added directly onto the strip, followed by addition of blocking buffers or staining reagents. During incubation, cells naturally settle to the bottom of the plate by gravity. Prior to washing the cells, ensure that the strip platform on Mini is adjusted to wash the desired column. The number of wash cycles and buffer flow rate can be easily customized. When incubation is complete, simply place the strip onto the platform, lower the fluidics head along using the lever, and commence wash. At the end of the wash, every well has a uniform volume of 25 microliters. The cells are now ready for collection to count and for the next step in sequencing. In the next couple of slides, I just want to quickly highlight some data we have run with collaborators, and it will complement the data that Bertrand and Antonino will present. The following examples show optimized Laminar Wash methods. We work with each customer to optimize the specific protocols. The efficiency of wash in sample preparation for proteogenomics and multi-omics is of high importance due to the sticky nature of nucleotide reagents and sensitivity of detection by sequencing. The speed and efficiency of Laminar Wash is illustrated in this example, where interleukin-7 is used to stimulate human T cells. They are removed from cells either by extensive 3 rounds of washing with large volumes of media using conventional centrifuge, or in a 5-minute wash by Laminar Wash. The cells were then allowed to rest for a specified amount of time, during which the elevated phosphorylated STAT5 protein gradually returned to resting values. The cells are subsequently fixed and stained for phospho-STAT5. T cells from which IL-7 was removed by Laminar Wash had faster decay of phosphostat 5 compared to Centrifuge, indicating that Laminar Wash can efficiently remove reagents in a rapid manner, as T cells are extremely sensitive to minute amounts of IL-7. In contrast, T cells that underwent Centrifuge had very slow decay kinetics, suggesting either IL-7 contamination or resulting from mechanical stresses from extensive centrifugation. There is an increasing drive to miniaturize assay and increase workflow throughput. Scarcity of biological samples further highlight the importance of low cell number retention. Our customers in gene editing and single-cell genomic fields requested information on washing low numbers of cells. We isolated naive T cells, then aliquoted the cells to 10,000, 50,000, or 100,000 cells per sample. The cells are subjected to 1, 2, 3 rounds of incubation and wash, consisting of simulated antibody staining, then wash either 2 rounds in centrifuge or 9 times on Laminar Wash. Laminar Wash maintained a high rate of cell retention relative to initial cell count throughout the different starting cell numbers. In contrast, samples processed using centrifuge wash had less than half of the initial cell numbers by the end of the third incubation and wash. Examination of the scatter plots also revealed significant decrease in the frequency of CD3 positive cells with each round of centrifuge wash. Compared to the initial frequency of more than 90% CD3 positive cells, centrifuge naive T cells had only a quarter of single-cell events being CD3 positive by the end of the third incubation and wash. The overlaid scatter plots showed that there is significant increase in forward side scatter low events, which are likely debris from centrifugal stress. Contrary to the belief that cell loss primarily stems from decanting after centrifugation, this result implies that mechanical stresses from centrifugation causes significant cell death and losses. Assays with isolated nuclei are particularly challenging, as nuclei are physically smaller than cells, making nuclei palette difficult to visualize after centrifuge wash. As nuclei have greater mass density than cells, they settle on laminar wash plates swiftly and can be washed gently without clumping or nuclei loss caused by centrifugation. Here, we isolated nuclei in bulk and proceeded to wash by either centrifuge or laminar wash. The nuclei are then stained by trypan blue and enumerated by cell counter. In duplicate wells washed by centrifuge, large clumps can be observed in one sample, while the second well experienced large loss of nuclei. This is in contrast to laminar wash nuclei wells that have consistent nuclear retention while remaining distinctly separated. This concludes my introduction of the Laminar Wash system and a few highlights of in-house and collaboration data. I'm now going to turn my presentation over to Bertrand from BioLegend. Bertrand, please.
Thank you for all attending this talk. I'm going to be going through some introduction and initial data for single-cell proteogenomics using the Curiox laminar flow washing system. So just to give you some background on proteogenomics for those of you who are not familiar with it, typically in flow cytometry in the past, many of you may know that when you stain with antibodies or fluorescent antibodies, you're limited by the visible spectrum. And so most people will run anywhere from 5 to maybe 30 antibodies at the same time for any given experiment. So a couple of years ago, BioLegend in-licensed a technology from New York Genome Center called CITE-Seq, and we started producing and selling DNA conjugates, where there is a DNA molecule with a PCR handle, antibody barcode, and a capture sequence, which are conjugated to the antibodies, which theoretically allow more than 1 billion unique barcodes based on a 15 base pair barcode. Obviously, we have reduced this barcode space to allow for about 5,000 unique barcodes in order to allow for errors during the sequencing of the library. So the general protocol for these proteogenomics assays are as follows. You stain your cells like you would in flow cytometry. You wash your cells, you would load on your single cell platform, in this case, the 10x Chromium. And then during the post-gym RT cleanup, you separate your library into what's called an ADT library or a gene expression library. Both these libraries are then pooled and then put onto a sequencer. Our product offering is as follows, with 3 formats for TotalSeq A, TotalSeq B, and TotalSeq C. TotalSeq A is basically just a poly-DT, or is compatible with any platform that has a poly-DT capture. TotalSeq B and TotalSeq C were specifically developed with 10x Genomics to be compatible with their V3, 3' V3 reagents and the 5' reagent kits, respectively. And then the output of the data looks exactly like flow cytometry. As you can see here, this is just an example of a very early experiment we did where we were looking at titration curves for varying, for a number of antibodies that we had conjugated. And so you can still see that there's good separation of the negative and positive populations, and they increase in signal as you would expect with increasing concentrations of antibody. Okay, so to move on to the Curiox Laminar Wash system, all of the data that you saw on the last slide were actually generated with centrifugation, which is our standard in our protocols and works very well. So one of the critical elements of this assay in particular is the cell washing, because if you don't wash well enough, your background can be quite high, as well as your signal for any given antibody in the pool. So what we recommend, which is a little bit more stringent than flow cytometry, typical flow cytometry staining is to wash 3 times with 3.5 mils in a fax tube. For some people, this may not work as well because they have low cell numbers, and so they'll try to use lower wash volumes to compensate. And so I've seen some people do 5 washes of 1.5 mils to get the same or equivalent staining efficiency. So basically, this ensures you have good signal to noise. However, you can imagine once you start processing many more samples, in our case, in the beginning, we might have been doing anywhere between 1 and 5 samples. Once you get past 10 or 20 samples, a lot of the samples are sitting there as you're decanting the buffer out of the fax tubes or whatever tube you're using. And so some of the samples may be sitting in buffer for longer than others, and this could affect the downstream analysis in the long run. So another thing that we've had a lot of requests for in terms of guidance is how to stain very low cell numbers, because you can imagine with very low cell numbers of anywhere between 50 and 100,000 cells, many people are very worried about losing their cells in the during the washing. We didn't actually run any experiments to investigate this yet, but Antonino, I believe, has some data that could be very useful for this case. So a lot of people who have these issues are isolating cells from tissue or have very rare cell populations that they're sorting out. And so this can be very problematic. Another thing is obviously many people have different protocols when washing by centrifugation, and so there can be very high user variation. For our experiment, we stained PBMCs with one of our large 277 antibody panel, which was pre-titrated and lyophilized. We also validated staining across our conjugate offering for this because we wanted to make sure that the staining patterns for all of the antibodies that we could detect in that panel were okay, and there wasn't any negative effect using the Laminar Wash. So for this comparison, we washed 1 sample by centrifugation using our typical 3.5 mil wash 3 times. And then for the other sample, we used the Curiox Laminar Wash system with a modified flow rate and cycle number. one of our initial experiments where we were using the recommended flow cytometry parameters of a flow rate of 5 and 9 cycles did not work very well. Like we said, this assay is very sensitive to insufficient washing, and so we had very high background in that experiment. So for this experiment, we upped the flow rate to 10, and then the cycle number was increased to 25. So the staining workflow is pretty simple. We block our cells typically in 25 microliters. We resuspend our lyophilized panel in 25 microliters, and then we add it to the blocked cells for a total volume of 50 microliters. Then we immediately transfer that 50 microliters to the Curiox plate. As was mentioned before, the reason why you want to put it immediately into the plate is you need some time for the cells to settle on the bottom of the plate, which is very convenient because the cell staining itself takes about half an hour, which is plenty of time for the cells to settle. Because of the nature of the proteogenomics assay and the desire to preserve the RNA in the cells, we incubate the cells while staining on a thermal tray from Thermo, which is put on ice. And so we make sure that we keep the plate and the cells cold while they're staining. Once the staining is done on the plate is transferred to the Curiox system for washing, and the whole process, at least with our settings, takes approximately 12 minutes, so it's less than 15 minutes for anywhere between 1 and 96 samples on a plate. And the data you get is actually pretty impressive. It's at least as good as centrifugation, as you can see. So in blue are the samples, or is a sample washed with the Curiox system, and on the bottom in orange red are samples washed by our centrifugation method. And as you can see, the results are almost the same, right? So you can still see very good separation of the negative and positive peaks, and this actually carries through to all of the antibodies that were used in the panel. So these are just representative results. So in conclusion, there's a very noticeable improvement in speed versus centrifugation in terms of processing time. It takes less than 15 minutes for us to use the Curiox for a very large number of samples, whereas it could take anywhere from 30 to 45 minutes for maybe 10 to 20 samples, or even much longer for many more samples. So this is very good for labs that have high throughput or need it for high throughput sample processing. For the data, the signal to noise ratio was just as good as centrifugation, utilizing our 3.5 mils and number of washes. For us, the ideal wash parameters for PBMCs at least was a flow rate of 10 and 25 cycles. Obviously, additional optimization may be needed for other sample types. So if you ever plan to use this system, I would suggest that you do some pilot experiments to make sure that it's working for your cell type. So although centrifugation results that we typically use and still use are very robust, the Curiox Laminar Wash can help with efficiencies when needed for high throughput labs and staining of low cell numbers. So whenever we use the Laminar Wash system, we always use it for large studies where we're trying to stain a large or high number of samples. Okay, so that's all for me. I'm going to pass it off to Antonino from Immunize.
Hi, everybody. Thank you for participating in this meeting, this webinar. I am Antonino from Immunize. What we do, at Immunize, we decode the immune system for better therapeutics, and we do so by interpreting the immune system by combining single-cell technologies together with artificial intelligence. And we're building a proprietary immune cell atlas that will allow for better therapies. This is our workflow, very similar to what Bertrand just showed. So we take samples, it could be PBMCs or other samples, we fold them, resuspend, removal of any contaminants. And if there are contaminants or the availability is lower than what we usually need for single cell, we enrich by sorting. And then after sorting, we label the cells similar to what Bertrand just described with CITE-Seq, and we do also cell hashing, which I'm going to describe soon as well. Then we do the single cell encapsulation using the 10x Chromium. Lab reconstruction, I have a multi-omic approach. We do both transcriptomics as well as proteomics or proteogenomics, TCR and BCR. Then we integrate the data and we apply our proprietary automated system to annotate the cells and finally do the analysis. The Curiox when we use it is after cell hashing and CITE-Seq labeling, so before encapsulation. And so what how cell hashing works? So this was actually a paper by Marlon back at the Genome Center. So the way the protocol works, you take your samples, so they are still in different wells. You stain them with TotalSeq antibodies for cell hashing, and the epitopes that they bind are the same for each one of your samples. And after staining together, so the samples are pulled together and then go through the 10x and single cell library prep. As you can see here, when encapsulation, you have cells. So some droplets are going to have one cell, but some are going to have multiple cells. So if they come from different samples, we take advantage of the cell hashing antibody barcode to enhance the multiplex identification, so enhance multiplexing, reduce batch effect, because you can imagine you can batch together different samples this way and reduce batch effect to sample processing or 10x plane used. And sequencing increases also reproducibility and reduces library preparation costs. So we first compare the cell recovery, and as you can see here in percentage, the Curiox really does a great job. So cells were washed similar to what Bertrand described, with 3 times the centrifugation. And for the Curiox, so when after sorting, especially, we end up with a higher volume, so there's a need to do a centrifugation step before putting into the Curiox plate. And then once there, we really have a nice recovery, and the Curiox has been great for samples that have a very low number of cells. That's when we mostly apply it to. So for more difficult samples. And if we have what we compare the Curiox to is like between a dishwasher and a wash machine. It's a dishwasher because your plates stay put, so your cells are going to stay put on the bottom of the Curiox plate. And it's like a washing machine because it washes away without centrifugation. That's more similar to the dishwasher, but you don't lose your socks in the process. Really, there's high retention. So these are some data from the cell hashing. If you look at them, we expect roughly a 20% of identifier of multiplex based on cell hashing itself. And the Curiox performs better when it comes to doublets. We're still investigating the reason for that, but it's very interesting results. And the number of negatives, so cells that were actually not stained, is also lower compared to the centrifugation. And number of singlets that we recover is pretty high in the 80%. So these are the numbers that we expect, and the Curiox really perform great. Regarding single cell RNA-seq, pre-filtering, the two actually perform pretty similarly in terms of number of genes that we recover with the 5' V1.1 from the next germ technology. And in terms of UMIs, when it comes to mitochondrial reads, it seems to be a bit higher in median for the centrifugation, but they're pretty similar. I mean, in both cases, the medians are similar, and there's a lot of cells that can be used. And with regard to the CITE-Seq, again, we use the TotalSeq C antibodies from BioLegend. The way it works, again, like Bertrand already described, we take patient samples, we label them with CITE-Seq antibodies. We usually use panel that go from 50 to 150 antibodies, gently and fastly prepared by Bertrand and his team. And once the samples are stained, we encapsulate and then do lab reconstruction. And we use this data in our multi-omic approach to annotate cells. And I'm going to show you here some UMAP. So it's a RNA-based UMAP. As you can see, this is actually from PBMCs. These are very nice clusters. I didn't annotate just for brevity of the webinar, but as you can see here, the cDNA, this high correlation between cDNA RNA as well for the protein. This is when we use the centrifugation. And the same with the Curiox. It's a very nice signal. When we look at CD8, this is for, again, for the centrifugation. You have also a nice signal here, and as well for the Curiox. It's a nice correlation here. I'm going to show some scatter plots. We see not really much difference between when it comes to the signal. This is only a couple of antibodies, I'm sure, but we see similar results for the rest 100 plus antibodies in our panel. So in conclusion, take-home message, so Curiox really helps retaining high numbers of cells, especially it's very, that's when mostly we use it, is for those samples have a lower number of cells to start with. And helps also with multiplexing multiple different samples together. And finally, Curiox centrifugation generates similar CITE-Seq results. So that's all from my side today, and ready to take any questions.
Wonderful. Thank you so much. Thank you, Chanying, Bertrand, and Antonino for this informative presentation. Really nice. We do have a few questions that have come in. Let's see here. The first one is for Antonino. Antonino, what made you switch to the Laminar Wash system?
So again, so we use both centrifugation and the laminar system. The laminar system really helps when it comes to, we switch to it, it's super helpful. And so first of all, the team is great, it's super responsive, Melvin and his team. And they help set up the system. So, and we use it when it comes to samples we already know have a lower number of cells, so lower quality, and for higher throughput projects. So we need really to push forward projects. That's also when we use the Curiox heavily. And the things that data are pretty comparable, we feel confident using it for this kind of situations.
Wonderful, thank you. To all, what is the optimal number of cells per well to maximize cell recovery? What's the upper end and lower end numbers?
So I can maybe, based on my experience, so that depends a lot on the cell type you work with. So also based on the cell type, I would, if this is a question later, maybe I would work with the team, or you can also play around with the flow rate as well as the number of cycles that you use. Each cycle is roughly 45 seconds, so that's fine. But coming back to the question, you can load, I think, up to 1 million cells, no problem, and retain a lot. We use it for low number of cells. I have more experience with those samples that have lower cells. If you input 50K, again, there is a centrifugation step where we might lose some, but you end up usually with 20 to 30K cells after that many cycles.
Okay.
So we tried lower, and is able to retain lower as well. And so you're gonna most likely end up with enough cells to load on the 10x.
Yes.
So maybe I can also supplement a little in this aspect. You know, in flow cytometry experiments, we have actually evaluated up to 10 million PBMCs and have quite comparable titrated retention, quite expected retention of 10 million PBMCs. We have not tried higher numbers. Presumably, we possibly can go higher. And as for the lower end, the image that you saw in the second slide of my presentation of the comparison between the centrifuge and laminar wash RFP cells, those were actually with 50 to 100 cells per well. So in theory, it can be extremely low and still have a high cell retention on the laminar wash.
Thank you. The next question, how can the wash parameter be optimized? Does it need a lot of downstream analysis?
So maybe I can, Bertrand, yourself some experience. So you definitely need to optimize that, so flow rate and as well as number of cycles. We did a couple of tests, some based on Bertrand experience, and some we just said that just changed. It's proprietary, so I'm not going to tell you how many cycles we do and the flow rate and other things that we do with the Curiox. But you have to play around a little bit, especially when it comes to background to signal ratio, and of course cells that you can retain. You have to monitor clamping and other parameters. You have to make sure that the buffer is the right one for your cells during the washing, that it's cold. So there are a few things that you have to play around. But then once you figure it out, it works pretty well consistently.
Great, thank you. Next question. In the peritoneal fluid after CLP, cecal ligation puncture, there are cells, bacteria, and debris from the stool. The laminar wash technology does wash away some debris, but the bacteria will settle to the bottom of the plate with the cells. How do you separate the bacteria?
Yeah, I don't know if Bertrand has experience. We usually don't work with this kind of samples. And I mean, what I would suggest you try to get rid of before putting into the Curiox. We usually use the Curiox for the labeling of antibodies. So I don't know how to answer that question. Maybe Bertrand can.
Yeah.
Yeah, we haven't had any experience with bacteria. I don't know if
Chen
has. Yeah.
Right. Thank you for the very interesting question. So we currently do not have a method to separate heterogeneous populations on the laminar wash, but I think you did bring out a pretty interesting application that perhaps we can look into in the future.
Excellent, good. Next question, would you say that you have no need to centrifuge since using laminar wash?
I'll take the first part, I guess. So maybe Antonino can comment later, but basically for us, we still do use centrifugation, like we mentioned before. It does depend on the cell type that you're doing, or the sample type. So I would say for most cases, it's convenient enough to do centrifugation if you're just doing a couple samples and you're just doing a pilot experiment. But for more high throughput studies or studies where you need more consistency within the data, I think that the Curiox is perfect for that. But I wouldn't say that it negates the need for centrifugation. There's always uses for centrifugation. Just really depends on what you're doing.
Great, thank you. This question is for Bertrand or Antonino. Have you tried to use the laminar wash system to scale down staining volume, cell number, and antibody mass, i.e. lower cell numbers, with the same cell and antibody concentration?
Antonino probably has experience with that, but I'll comment on the concentration part. So normally, at least for this assay, the most important part is the concentration of antibodies. So whether or not your cell number is higher or lower, you just need to make sure that your concentration of antibodies is the same. So for example, for our pre-made panels, they're always meant for a specific reaction size. So in our case, it's 500,000 cells in a total staining volume of 50 microliters. So if you plan to stain 250,000 cells, for example, you would need to do it in a total staining volume of 25 microliters. So the most important part is the concentration here. You can, of course, stain a lower number of cells in the same volume, but that's, you'd have to test it out for yourself. But Antonino, I think you guys do lower cell numbers already.
Yeah. So that's a fair question, and yeah, that's also what we played around with. So you can definitely reduce volumes and cell numbers and everything else. But there's a lot of optimization that needs to be done. But yeah, it can be done.
Thank you. Next question. So did you run a whole CITE-Seq experiment, sequenced, and then checked the ADT signal-to-noise ratio to optimize the wash parameters? Or did you optimize it with pilot FACS experiments?
For us, we went all the way to the end because the final output was going to be for CITE-Seq. One of the things that we did notice, like I mentioned, was that the CITE-Seq is much more sensitive to insufficient washing, which is why we had to analyze it by sequencing. So for us, because the endpoint was sequencing, we did have to look at the data for that. But we can obviously, our parameters are a good starting point, and I would definitely do a pilot study to verify whether or not that works for your sample type.
Yeah, same here. We went full in with the single cell. So when you optimize, you might want to go directly with the CITE-Seq and full pan and the full protocol, just because the washing is different. I mean, also in the flow, you usually work with higher volumes, so washing is much easier there. And you want to really be careful with the background that you get from the CITE-Seq protocol. That's why I would suggest to invest some money and time on that before going full steam.
Mm-hmm. Thank you. A question for you, Antonio. Does ImmunAI offer services for CITE-Seq?
Yeah, we definitely do use CITE-Seq pretty much regularly. And we have a couple of panels that we're going to consolidate immunological panels together with Bertrand from the TotalSeq, but yeah, that's what we offer.
Good, thank you. This is for Bertrand. What kinds of samples does BioLegend recommend that people use Laminar Wash instead of a centrifuge?
Well, I mean, we've only tested PBMCs. I would imagine any type of normal cell line. I think, correct me if I'm wrong, Antonino, but I think you guys do tissue. Have you guys done dissociated tissue at all?
So it was pre-dissociated, and we did the sorting, but yeah, it worked very well. CITE-Seq from TILs was amazing as well.
Yeah, I would say we don't have much experience with that many different cell types because we just don't have access to them. But I think Antonino has a better idea of real-world samples more than we do. So I think, like you said, the dissociated tissues can work just as well.
Thank you. Next question, this might be for you, Chen-Ying. How long does it take for cells to settle on the Laminar Wash plate?
Right. That really depends actually on a few different on a few parameters. In general, cells, anything that is denser than buffer will eventually settle. So anything with nuclei, whether it's cells, bacteria, yeast, they all settle in a pretty short amount of time, about 20 to 30 minutes if you're starting with, say, 1 million cells in each sample. And of course, if you have less or more, and if you're working with cells that are less dense in nature, those can also change the settling time, alter the settling time a little bit, but in general, within 20 to 30 minutes, 1 million PBMCs is pretty safe.
Maybe I can add something. You can even look under the microscope. You can really see the cells sitting down on the flat surface of the Curiox plate. So when you play around with your cells, also pay attention to when they sit down, and you can monitor that over time.
Mm-hmm. Absolutely. Thank you, Antonino.
Mm-hmm. And our final question is for Antonino. What made you switch to the Laminar Wash system?
Maybe at a similar question earlier. Yeah, the throughput and then the ability to retain low number of cells. So we work with different collaborators. They have different types of cells, and so the quality is very variable. So the Curiox really helps for those samples where the quality is lower than what we usually work with.
Thank you.
And yeah, that's one of the main reasons.
Thank you to Chanying, Bertrand, and Antonino. Excellent presentation, which will be available on our website. And this is the end of the webinar. Thank you.